Have you ever actually stopped to wonder what is physically happening inside your head when you learn something new? For decades, science basically treated the human mind as this impenetrable black box, just a total mystery. But today, well, we know so much more. Welcome to This Explainer. I'm your host, and I'm absolutely thrilled to be your guide as we crack that black box wide open. Today, we're exploring the biological roots of intelligence and learning. We're going to look at the exact physical mechanisms that turn fleeting, everyday experiences into lifelong knowledge.
Okay, let's dive into this. Here's our roadmap for today. 1. The behaviorist black box. 2. Donald Hebb's radical idea. 3. The Hebbian triad. 4. Molecular coincidence detectors. And finally, 5. 2 phases of memory.
All right, kicking things off with Section 1, the behaviorist black box.
You see, mainstream psychology used to look at the mind with a pretty dismissive view. They championed behaviorism. To them, the mind was literally an unobservable black box. Learning was viewed merely as a simple reflex loop. An external stimulus goes in, a response comes out, and whatever happens beneath the skull, well, they deemed that just an irrelevant mystery. But then, in 1949, a Canadian neuropsychologist named Donald Hebb came along and absolutely shattered that paradigm. He argued for the physical mind. He made this massive, paradigm-shifting realization that mental concepts aren't just abstract illusions. They have explicit physical representations within the distributed biological networks of our brains. Basically, individual biological neurons actually generate human intelligence.
Moving right along to Section 2, Donald Hebb's radical idea.
Hebb was essentially the detective who found the very first clues to crack open that black box. And he probably did it because he had a highly unconventional background. If you look at his academic evolution, he didn't start in a lab at all. In the 1920s, he was actually an elementary school principal focusing on pedagogy. He then moved on to get his PhD studying the visual system under Carl Lashley. But the real turning point was his 1937 stint at the Montreal Neurological Institute. Here, he collaborated with the pioneering neurosurgeon Wilder Penfield. Hebb systematically evaluated human intelligence before and after massive surgical lesions, and he observed an incredible anomaly. Patients who had massive unilateral removals of their frontal cortex didn't suffer catastrophic drops in their standard IQ scores. I mean, how could that be? He later drafted his masterpiece at the Yerkes Primate Labs, but this clinical reality in Montreal, it changed everything.
So to figure out what was actually happening with those surgery patients, Hebb invented the McGill Picture Anomalies test. What it revealed were subtle, diffuse cognitive deficits, rather than some kind of total intelligence failure. And this brilliantly illustrates how cognitive processing actually emerges from distributed, resilient, highly interconnected networks. Intelligence doesn't just sit in one isolated center of the brain, right? Nor is the brain just some undifferentiated mush. It's a highly networked system.
Which perfectly sets up Section 3, the Hebbian triad. Now that we understand the whole brain as a network, let's look at exactly how those connections form. To completely replace behaviorism, Hebb proposed 3 interconnected neuropsychological pillars. These act as a direct bridge between your subjective cognitive experiences, your thoughts, and tangible neurophysiology. They are synaptic plasticity, Hebbian cell assemblies, and phase sequences. Let's break them down.
First up, synaptic plasticity. Basically, when a presynaptic cell persistently fires into a postsynaptic cell, it causes structural or metabolic growth in either one, which increases their driving efficiency. The key takeaway here? Learning doesn't mean growing entirely new anatomical pathways from scratch. It actually means physically upgrading and strengthening the pathways we already have.
Next, we have Hebbian cell assemblies. When you experience a stimulus, a specific group of neurons is repeatedly activated together, and they sort of weld into a cohesive unit. The crucial point here is that this unit becomes capable of self-sustained reverberating neural activity, meaning the electrical signals continue to circulate long after the external stimulus is gone. This reverberation bridges the temporal gap between stimulus and response. Guys, it is the literal, physical substrate of an independent thought. No way, right?
So let's move to phase sequences and see how this all builds. Phase sequences are temporally and functionally linked networks of those individual cell assemblies. When one assembly activates, it sequentially ignites the rest of the sequence through learned associations. This sequential ignition physically maps the continuous fluid stream of human thought. It's exactly how one idea naturally flows into the next. And here is exactly why this matters to you. Think about the difference between childhood learning and adult learning. Childhood learning is this slow, painstaking, incremental buildup of foundational cell assemblies and phase sequences. But adult learning, which is what you're doing right now, is incredibly rapid. You aren't building from scratch anymore. You're performing a real-time, insightful Gestalt rearrangement of decades of established neural structures just to adapt to complex new data. You are literally rewiring your existing network.
Okay, let's zoom in even closer. Section 4, Molecular Coincidence Detectors. Let's go right into the microscopic synaptic cleft. Decades after Hebb's theory, modern molecular biology actually validated his insights through the discovery of long-term potentiation. At the cellular level, building a memory relies on the elegant coexistence of two essential postsynaptic receptors. First, we have AMPA receptors. These are your standard gating channels. When glutamate hits them, they rapidly let sodium in for a quick depolarization. Second, we have NMDA receptors, and these are the stars of the show. They are molecular coincidence detectors, but under normal conditions, they are physically blocked by a magnesium ion. Nothing gets through.
So how do we get that magnesium block out of the way? Well, this is where the magic happens. Two independent conditions must hit simultaneously. Step 1, the presynaptic activity must release glutamate. Step 2, the postsynaptic membrane must fiercely depolarize via intense AMPA receptor activity to electrostatically kick out that magnesium ion. To expel the block, both conditions absolutely must occur at the exact same time. This physically proves Hebb's rule of causal correlation.
And once that magnesium is kicked out, we get the ultimate trigger. Unblocked NMDA receptors suddenly allow a heavy influx of calcium ions right into the postsynaptic cell. I really want to emphasize this. This heavy rush of calcium ions is the ultimate biochemical trigger. It's the master key that unlocks actual structural change in your human tissue.
And that brings us to our final zoom-in. Section 5, 2 phases of memory.
Let's explore the magnificent biochemical cascade that this rush of calcium sets into motion. But first, a quick question for you. Why do you remember a vivid childhood memory forever, but you forget a phone number in like an hour? It's something we've all experienced, right? Well, the answer lies in the data. Calcium actually divides memory into 2 distinct phases. Early phase lasts just 1 to 4 hours. It is completely independent of new protein synthesis. It just uses what's already there. Calcium triggers local enzymes, like CMK2 and PKC, to simply modify existing AMPA receptors. There is absolutely zero structural remodeling here. But late phase? That endures for days to years. It requires the synthesis of entirely new proteins. Signals travel all the way down to the cell's nucleus, kicking off a chain reaction of new kinases like ERK and MAPK to CREB. The result? Actual physical, morphological changes. It is a spectacular biochemical chain reaction. To distill all that data down, early phase, or ELTP, gives you temporary biochemical alterations to existing synapses just to hold immediate data. That's how you hold the phone number temporarily. But late phase, or LLTP, engages permanent structural remodeling. We're talking about synaptic knobs physically enlarging and entirely new dendritic spines sprouting. LLTP literally alters the geography of your brain to wire in permanent expertise.
I want to summarize the core takeaway of this explainer with this really powerful thought. To build deep, domain-specific expertise or lifelong memories, your brain must physically synthesize new structural proteins to alter its geography. We've traveled from the whole brain down to the network, into the synapse, and to the calcium receptors. But ultimately, all this complex biochemistry grounds us right back into the beautiful reality of how human intelligence evolves over a lifetime. Abstract concepts literally become structural neurobiological realities.
I want to leave you with one final thought. Learning isn't just something that happens to you. It's a physical transformation within you. As you process the information in this explainer, right at this very moment, your NMDA receptors are unblocking. Calcium is flooding in. So I have to ask, how are your own neural networks physically rewiring and sprouting new dendritic spines right now? Your brain structure is physically transforming simply by having explored this topic. Thank you so much for learning with us, and keep rewiring those networks.